much for inviting me here, and thanks to Srujan and other team members for inviting me. And thanks for the kind introduction. I hope this comes. I mean, as he introduced me, I'll be talking about a new class of small RNAs, which came as a surprise to us. In fact, the discovery was very serendipitous, where we are trying to understand what small RNAs do, and we landed up identifying a new class of small RNAs called tRNA-derived fragments. So what is the focus of my lab? My lab is interested in looking at how stem cell state transitions happen. How stem cell state transitions happen. The stem cells perceive a signal, and they have to get out of the stem state and starts differentiating. So what are the mechanisms that drive the stem cells to emerge from the stem state and differentiate? That's the questions we are asking. So the first line of gene expression regulation that should happen, it should happen at the level of proteins, because you need to bring down the proteins, which could be enzymes, and it could be structural proteins or transcription factors. You need to bring down these proteins so that the stem cells emerge from the stem state and starts differentiating. And this protein repeatide downregulation can happen in two ways. One is I degrade the protein, and the other is, as I'm degrading the protein, I can't keep making the new protein. So I have to also regulate the protein synthesis, and that is what we are interested in looking at, how protein synthesis regulation is happening. And we use two interesting model systems. One is planaria, which is a regenerative invertebrate model, and other is, of course, the embryonic stem cells, which we know are pluripotent in nature. So why translation, and how many different ways we can regulate translation? The translation can be regulated at three different positions. One is initiation, where the ribosomes are loaded. Ribosomes elongate, it's called elongation, and the ribosomes separate off, and that's called termination. So, and one of the biggest bottlenecks of translation in terms of regulation and energetics is the initiation. So we looked at a lot of proteins here. We looked at a poly-A binding proteins that bind to the poly-A tail, and that poly-A binding proteins are critical for recruitment of initiation factors. And there are many different poly-A binding proteins, and these different poly-A binding proteins, the way they recruit ribosomes, regulate the translation and regulate how the cell fate decisions are made. And when I talk about poly-A binding proteins, we also look at poly-A tails, because every eukaryotic mRNA has a poly-A tail, and a poly-A binding protein binds to it and regulates. So if I change the length of a poly-A tail, I can change the fate of the mRNA, thereby I can regulate how these stem cells are either differentiating into specific lineages. So this is all fine, and we are also looking at microRNAs that bind to the 3' UTRs and regulate translation. And from last 5-6 years, we are interested in understanding ribosome. Ribosome for a very long time were thought as a monolithic structures that sit and make polypeptides. But we know that now the ribosomes act as a regulatory hubs. And if you look here, and we call it as a ribointeractome, the ribosome assembly itself is a huge energy-driven process, and by controlling how the ribosomes assemble, you can control the stoichiometry of ribosomes and thereby regulate protein synthesis and regulate cell fate decisions. And we are looking at specific helicases that are important for ribosome assembly. We also look at specific rRNA expansion segments. These are very specific to mammals, and these rRNA expansion segments recruit the ribosomes and regulate how ribosomes actually elongate on an mRNA, and thereby regulates the outcome, which is the proteins here. And the third one is post-transcriptional and post-translational modifications. The ribosomes the rRNA is heavily post-transcriptionally modified. They are methylated, acetylated, and the proteins of the ribosomes also undergo post-translational modifications. By changing these modifications, I can regulate how the ribosome is bound to the mRNA. The third part, that is what I'm going to talk about, is the tRNA-derived small RNAs. And what I'm going to talk about is what are they and what are their functional roles. So, what are tRNAs? And this discovery started happening in as late as 2018, and it's not 2018, actually it's 2010. That's where the discovery started coming up. We know that the tRNAs carry amino acids, and they are important for polypeptide chain formation. Imagine this fundamental molecule can be broken down into pieces, and each piece can go and regulate specific aspects of these cellular processes. And one key molecule I will be talking about today is called 5' turfs, which are 25 to 34 nucleotide in fragment. And when they discovered this 5' turfs or 5' tRNAs, they found that they are important for when you induce the cells with stress, they are upregulated, they have an anti-cancer property, they can be used as a biomarkers. What we are going to talk today is its role in maintaining of pluripotency. So how did we discover this? So we were working on ESCs, and this is work done with Ramanuj and Srikala, who are now in Glasgow. So what we did is we took the ESCs and we treated the ESCs with retinoic acid. When we do that, the ESCs differentiate into the fate of neuroectoderm. And when you actually treat with the Wnt inhibitors, it actually becomes the prime state or a ground state. So the simple question we asked is, what are the small RNAs that are upregulated during the differentiation within 48 hours of retinoic acid treatment and in the prime state? That's the question we asked. And very interestingly, what we found is we found this peak, which is red in color there, that starts emerging, and this peak is at the size of 25 to 34 nucleotide. And this peak goes up as the stem cell starts differentiating. And that's the histogram that is shown there. And the question we asked is, what is those 25 to 34 nucleotide species? And it turns out that all these species actually map to the tRNAs. That means the tRNAs are being processed into 24 to 35 nucleotide. The first thing that occurs to our mind is, if these are the products of tRNA, this could be just a degrading product, right? So we ruled out that, because when we take these small RNA species, which are the 5' tsRNAs, and we map it to the tRNA, all of them start from the 5' end, and they go all the way down and ends right before the anticodon arm, suggesting that if that's a degradation product, it should emerge from the whole tRNA. But that's not what is happening here. They are only coming from the 5' end of the tRNA, right? And this is a qPCR validation showing that, suggesting that this is just not a degradation product, there can be a functionality to it. So how do we show the function? In order to show the function, we did two experiments. First, we took ESCs, we added retinoic acid, right? And we added an antisense oligose of the tRNA. If we add the antisense oligose, what we assume is it blocks the tsRNAs and prevents the ESCs from differentiating, right? And how do you measure the ESC activity? By alkaline phosphatase activity there. And that is how we measure whether it's in an ESC state or it's in a differentiated state. That's what we asked. And what we found is, when you take ESCs and add retinoic acid, the ESCs differentiate, alkaline phosphatase activity should come down naturally, but when we add antisense oligose, we see increase in the alkaline phosphatase activity, suggesting that if you block the tsRNAs, you can keep the stem cells in a stem state for a very long time. The second question we asked is, what happens when we add the mimic? For example, if you take a LIF ESC here, LIF maintains ESC in a pluripotent state. So now if we add the mimic, that means we are increasing the concentration of the tsRNA, and we ask the question, what happens to ESC? Even in the presence of LIF, the ESC starts losing its alkaline phosphatase activity, suggesting that the mimics are, if you increase the concentration of the tsRNA, you push the ESCs from a pluripotent state to a differentiation state, suggesting that the small molecules that we have identified, which we call as 5' tsRNAs, has a functional relevance to it. So, great. The second question we asked is this tsRNAs associated with ribosomes? The answer is yes, they are associated with ribosomes. They are associated more with the polysomes compared with the monosomes and the subunits of the ribosome. Does they inhibit translation? And what we did is we did this in vitro translation assays where we take rabbit reticulocyte lysates, we add GFP mRNA, and we add the mimic and the scramble, and we asked for how much GFP is being synthesized here, and we saw that when you add the mimic, the GFP levels actually go down, suggesting that it inhibits the translation. But this experiment is little misleading. This experiment suggests that you are blocking the whole translation, but the answer is that's not true. If it blocks the whole translation, the ESCs will not differentiate to start with. That means something else is happening. To understand what is happening, what is the function of this tRNAs, what we have done is we have done this interactome studies. The way we did these interactome studies is this. We took ESC leaf, we treated them with retinoic acid. After 48 hours, we took the extract. We used bioplated mimic, which is a tRNA, and we did the pull downs. We did the proteome analysis, we did the transcriptome analysis to identify what are the interacting partners of the tRNA. Since it's a published data, I am just summarizing the results here. What we found is when you add tRNAs and when you pull the tRNAs bind to the pluripotency factors when you treat them with retinoic acid, suggesting that the tRNAs are actually inhibiting the transcripts, the translation of the transcripts that are essential for maintenance of pluripotency, thereby pushing them to differentiate. The second part is we asked what kind of proteome is interacting with tRNA. We found that there are two interesting proteins that interact with tRNA. One is called IGF2BP1 and YBOX. These two are RNA binding proteins that are important to stabilize c-MYC. I will come to that in a short while. And we have also shown that IGF2BP1, which is an RNA binding protein, is critical for the maintenance of stability of c-MYC there. And the third is when I block the tRNAs, the c-MYC, which is important for cell cycle regulation, actually gets destabilized. So based on these observations, this is the model we propose. The model is this. Under ESC conditions, the two RNA binding proteins, IGF2BP and YBOX, binds to c-MYC mRNA. They stabilize c-MYC mRNA and load the c-MYC mRNA into the ribosome, and the translation happens. When I take ESCs and treat them with retinoic acid, what happens is the tRNAs that are upregulated upon retinoic acid treatment goes and binds to IGF2BP1. They dislodge IGF2BP1 from the c-MYC mRNA. As a result, the c-MYC mRNA gets destabilized and subsequently gets degraded. So it's a very interesting way of regulating the cell cycle genes and also pluripotency factors, and the regulation is happening by dislodging the proteins from the mRNA, thereby making the mRNA unstable, and the mRNA gets degraded and the protein levels go down. So this is the model we have proposed. The next question we asked is, if this is true, how is the biogenesis of tRNAs happening, right? And that's a holy grail now to understand how our tRNAs are being made. We know that there is an enzyme called endoribonuclease that takes the tRNA and cuts into the 5' tRNA. When the angiogenin cuts the tRNA into 5' tRNA, they add this 2', 3' cyclic phosphate groups. So when you add that, in order to clone the 5' tRNA for your studies, I need to do the kinase treatment and then clone. But when we are initially studying the 5' tRNAs, we didn't do this kinase activity. What we did is, we took the 5' end and we directly sequenced it, suggesting that there are actually two pools of tRNA. One pool is angiogenin dependent, the other pool is angiogenin independent. So the question we are asking is, what is this angiogenin independent pool? One pool is coming from an angiogenin which is cutting the tRNA to make tRNA. The other pool is an endoribonuclease, which we don't know what it is also cutting and generating tRNAs, and their expression in a stem cell is inversely proportional. When angiogenin is low, the other one goes up, right? So the question is, what is the other one? In order to address this question, we went to this invertebrate pluripotent model, which is called Chlamydia mediterranea. And what is this model? It's a flatworm, and it has immense regenerative abilities. They grow in fresh waters. You can take that little species, you can cut them into small fragments, and each fragment can regenerate back into a complete animal, right? And they have this bag of stem cells distributed all over the animal, and they are pluripotent in nature. So if you want to understand pluripotency, this animal serves as a beautiful model to understand what is pluripotency, right? So we use this animal and we ask three questions here. One is, does these animals have tRNA? And are the tRNAs expressed in stem cells? And how is the biogenesis regulated? I will just summarize the results here. In order to address these three questions, what we did is, we took planaria, we chopped planaria into tiny pieces, and also we took the regenerating tissue that is white in color there, and we have done an extensive small RNA sequencing, and this is the summary of that result. We identified multiple species of tRNAs that are specifically expressed in planarium model here. One is the 5' tRNA, which is 25 to 30 nucleotide in length. The other is we call as TERFs, and these TERFs are only 18 to 24 nucleotide, and one TERF is generated from the 5' end, and other generated is from the middle region, which is called I-TERFs. So don't worry about that part of the data here. Let's worry about the 5' tRNAs. These are the same tRNAs we have identified in the pluripotent stem cells, or mouse embryonic stem cells. The question is, how are those 5' tRNAs generated, and what is endoribonuclease? First, we thought that this should be an angiogenin, but it turns out that there is no angiogenin in planaria. So we did an RNA screen of endoribonucleases, and we found a specific protein called PV protein. The PV is nothing but an organate family of proteins. They are same as the organate family of proteins that are essential for microRNAs. And there are three PVs in planaria called SMEDV1, 2, and 3. All the PV proteins are expressed in the stem cells of planaria, and the knockdown of 2 and 3 leads to a defect in the stem cell maintenance, right? So this is all good. We knock this down, and the tRNA levels go down, right? But the conclusive experiment is to show that the PVs are interacting with these tRNAs. So for that, we have used this data that was generated by Kim et al. From planaria, where what they have done is they took these three PV proteins, they have done the pull down and asked what kind of small RNAs are associated with it. But very interestingly, if you look at that data, maximum data what you see is no annotation. They couldn't annotate what the small RNAs are when they published their paper in 2020, because they don't have the idea of tRNAs at that point. So what we did is, we took that data and asked, are they coming from tRNA or whether they are coming from tRNA? You can see that the SMEDV2 and 3 has the highest association of these proteins with the 5' tRNAs. This shows that in addition to angiogenin, the PV proteins can also cut the tRNA to generate tRNA. So the next argument is, this is all in planaria. Is this true with humans, right? Or mouse? So to that, you can see a Western blot here. You can see that the angiogenin is highly expressed in the ESCs, in the lift conditions, right? And as the stem cell starts differentiating, we identified a specific PV protein called PVL4 that is highly upregulated as the stem cell starts differentiating. LIF is a stem cell, and if I put in retinoic acid, they start differentiating. As they differentiate, the PV signal goes up. So we are currently doing knockdowns to understand its role in tRNA generation. But what we hypothesize here is this. You have two pools of tRNA. One is angiogenin dependent, other is angiogenin independent. We think that angiogenin independent tRNAs are coming from the PV proteins. And the PV derived tRNAs we think are critical for the differentiation. And to maintain the ESCs, it comes from the angiogenin dependent tRNAs. So now you have two ribonucleases. One is, and both of them are cutting tRNA, and both of them are generating 5' tRNAs. The big question is, how are they regulated, right? So for instance, if you look at this diagram here, or figure here, you can see that there are pool of tRNAs. Only very few are generating into a tRNA there. And if you look at a single pool, if I have 1,000 molecules of tRNA, the question we asked is, how much of the tRNA is being processed into tRNA? The answer is only 1 to 5% of the tRNA is being processed into tRNA. that means there is a huge regulation that is happening, and that regulation is critical for generation of tRNA, right? So how is that happening? The answer actually lies in the modifications of tRNA. Another 2 minutes I will close it. So if you look at modifications of DNA and RNA, one of the molecules that is heavily modified is the tRNA. tRNA has 40 different modifications, or have 40 modifications, and they have almost 22 different modifications, right? So the question we asked is, does these modifications help us to understand how the regulation is happening? There is an interesting paper that came from Blanco's group. What they have shown is the angiogenin can cut an unmodified tRNA to generate tRNA. But when there is a methylation of cytosine right at the anticodon arm by a protein called NSEN2, which is a methyltransferase, the angiogenin fails to cut. As a result, the tRNA is not broken down into tRNA. In a way, the answer is yes. The modifications of the tRNA are regulating how a tRNA is being processed into tRNA. Now the question we asked is, what about PV, right? In order to understand that, what Anusha, a postdoc, did in my lab is, she took all the modifying enzymes and did an RNA screen. The first one is, you are seeing, is pseudouridine, M5 cytosine, M1 adenosine, M3 cytosine, and what you see is a bunch of enzymes. What she has done is, she has done an RNA screen of all these enzymes, and the first question we asked is, does it affect stem cells? And what we found is, the PUS3 has a defect in differentiation, NSEN2 has a defect in maintenance, and TRM6 has a defect in maintenance. So you can see that every knockdown of this enzyme has a specific defect on whether the stem cell is being maintained or being differentiated. The next question we asked is, what is happening to tRNAs? And what we found is, when you knock down PUS3, which is a pseudouridine synthase, what happens is the 5' tRNA levels go down dramatically, suggesting that this modification is important to generate tRNA, right? So the question we asked is this, and we know that when we knock down PV, the tRNA levels go down. The question we asked is, does these two match or not? So what we did is, we took all the downregulated tRNAs from here and from the PV knockdown, and we asked how many of them are common between both, and we found that 90% of the tRNAs that go down in PUS3 are same as the PV knockdown. Based on this data, what we propose a model is this. You have a tRNA, and PSU3 is an enzyme that adds pseudouridine group to the D loop of the tRNA. By adding the pseudouridine group, it allows the PV to go and cut the tRNA to generate tRNA. So now if I knock down the PUS3, you have an absence of pseudouridine group there. As a result, the PV fails to go and bind to the tRNA. As a result, it doesn't generate the tRNAs. Rather, the tRNA fails to be degraded into tRNA. So this is very interesting, right? In one case, a modification is allowing the processing. In other case, the modification is blocking the processing. So we are actually coming out with a theory that these modifications of tRNA, which we don't know what they actually do, play an interesting role in generation of these small RNA fragments, which we call as tRNAs. So with that, I will summarize the result here. So we have identified two classes of tRNAs, and one class comes from angiogenin, which is an endoribonuclease, and that class is important for the maintenance of pluripotent stem cells. The other class of tRNAs are coming from another endoribonuclease called PV, and they are essential for the differentiation of pluripotent stem cells into a differentiated cells. The question we are asking is, if we reverse this, can we push the differentiated cells, or can we reprogram the differentiated cells to become a pluripotent stem cells? In other words, what we are asking is, can the tRNAs be used as a molecular markers or molecular fine-tuners for changing the cell states, right? And that's the question we are asking now. And with that, I will stop the talk. And and the initial part of the work was done by Srikar and Vairavan, and Anusha was a postdoc, she left now, and she worked on tRNA modifications and their role in generation of tsRNAs. And we have done that work in collaboration with Ramanuj and Srikala group. Thank you very much, and I'm happy to take questions.